Malnutrition is an ongoing crisis projected to affect over 600 million people by 2030. The World Health Organization defines malnutrition as a deficiency, excess, or imbalance in a person's intake of energy and nutrients. The more recognized form of malnutrition is undernutrition, manifesting as a loss of muscle mass due to insufficient energy and nutrient intake. Short term, it's known as wasting, and long term, it's known as stunting. In 2024, 23.2% of children under the age of 5 experienced stunted growth, and 6.6% experienced wasting. The less severe form of malnutrition is overnutrition, which is a surplus of energy and nutrients that leads to chronic disease, but is rarely immediately detrimental to biological function. Addressing this is why Sustainable Development Goal 2.2 is so critical. It aims to end all forms of malnutrition by 2030 with a focus on the most vulnerable groups, including young children, pregnant women, and the elderly.
Nutrients are substances that the body needs for biological processes that it either cannot synthesize or cannot synthesize enough. Therefore, they must be obtained from our diet. They can be categorized as either macronutrients or micronutrients. Macronutrients are organic molecules which include carbohydrates for energy storage, proteins for muscle building and enzymes, and lipids for cell structure and hormone production. Micronutrients can be inorganic minerals or organic vitamins which act as regulatory coenzymes and cofactors within the chemical reactions in our body. While imbalances in these nutrients have a general negative impact on bodily function, individuals under major metabolic stresses are highly susceptible to specific deficiencies. This leads to young children, women, and the elderly at the greatest risk of malnutrition. The SDG Target 2.2 specifies in its first 2 indicators that stunting and wasting are major concerns in children under the age of 5. During this rapid growth period, cells require a large amount of energy, meaning they need a large quantity of macronutrient-rich foods. When deprived of this, their growth is stunted and, in extreme cases of wasting, suppresses their immune response and healing, leading to metabolic failure. The third indicator of malnutrition outlines the prevalence of anemia in women, specifically when menstruating, pregnant, or lactating. Anemia occurs due to the lack of iron needed for hemoglobin production, and when combined with blood loss causes reduced red blood cells and therefore decreased transportation of oxygen around the body. For the elderly, protein and vitamins are needed to prevent sarcopenia, better known as muscle death, and support their immune response.
CRISPR-Cas9-based genome editing has become the most prevalent approach to improve crop varieties. The technology works by inserting a guide RNA into the cell, which recognizes the target strand of DNA to be cut for editing, in order to replace or knock out the gene. This allows biologists to alter a cell's function and improve the stress resistance and nutritional quality of crops for applications in countries more severely impacted by malnutrition. For example, researchers have used the technology to edit potential genes causing susceptibility to MLN disease in maize crops, with the edited lines now ready for field trials to test their resistance under real-world conditions. With the ability to destroy 20 to 100% of the crop, this could be applied to increase food production and thus potentially reduce malnutrition, stunting, and wasting. Further, sorghum is regarded as the second most significant crop in Africa, despite having a low protein digestibility due to low levels of amino acids. Studies reveal a sorghum mutant genotype with low levels of the alpha-caffeine storage protein has increased levels of the amino acid lys. The altered protein structure makes it more accessible to gastric proteases and increases protein digestibility. CRISPR genome editing has been used to create sorghum varieties with this quality, demonstrating the potential to improve the nutrition content of staple crops. The poor iron bioavailability in staple foods like cereals is one of the major etiological factors for the high prevalence of iron deficiency anemia in developing countries. Developing nutritionally enriched crops is thus a potential avenue to address vitamin-deficient conditions such as anemia.
To summarise, limited funding, inadequate support and restrictive policies mean GM crops have failed to deliver nutritional benefits to populations. In countries like Uganda and Nigeria, weak regulatory systems and slow approval rates further block progress. However, research in Australia is being done, where in 2024 5 GM crops were approved. As well as the oversight by monitoring agencies like the FDA and EFSA are building trust by providing data and transparency. Growing acceptance has led to increased investment, such as the Bill and Melinda Gates Foundation's funding of CRISPR research for sickle cell disease and HIV. CRISPR-Cas9 holds strong potential to improve crop nutrition and supporting Sustainable Development Goal 2.2. However, barriers need to be overcome for CRISPR-Cas9 GMOs to reach the populations who need it most.